Welcome to our deep dive into multiple sequence alignment, or MSA. In the world of bioinformatics, MSA is a foundational technique used to compare, analyze, and interpret biological sequences such as DNA, RNA, or proteins. By aligning multiple sequences together, researchers can identify regions of similarity and difference, which often reveal evolutionary relationships or functional similarities. Throughout this video, we'll explore what MSA is, why it's important, and how it's applied in real-world research. Let's begin by understanding the basics of sequence alignment and how MSA fits into the bigger picture. Before diving into multiple sequence alignment, it's essential to grasp the concept of sequence alignment itself. Sequence alignment is the process of arranging sequences of DNA, RNA, or proteins to identify regions of similarity. These similarities may indicate functional, structural, or evolutionary relationships between the sequences. Alignments can be pairwise, involving just two sequences, or multiple, involving three or more. The goal is to line up the sequences so that similar or identical characters are in the same column, making it easier to spot conserved regions and mutations. Lining multiple sequences at once offers a broader perspective than pairwise alignment. By comparing several sequences together, researchers can detect conserved motifs, evolutionary patterns, and functional domains that might be missed when looking at just two sequences. Multiple sequence alignment is especially valuable in studying gene families, protein domains, and evolutionary trees. It helps in predicting the function of unknown genes and understanding the evolutionary history of organisms. The insights gained from MSA are crucial for fields like genomics, molecular biology, and evolutionary studies. Multiple sequence alignment relies on several core principles. First, it seeks to maximize the alignment of similar residues across all sequences, highlighting conserved regions. Second, it introduces gaps where necessary to account for insertions or deletions that have occurred over evolutionary time. Third, it uses scoring systems to evaluate the quality of an alignment, balancing matches, mismatches, and gaps. These principles ensure that the resulting alignment is both biologically meaningful and computationally optimal, providing a solid foundation for downstream analyses. MSA, the sequences being aligned can be DNA, RNA, or proteins. Each type of sequence presents unique challenges and opportunities. DNA and RNA alignments are often used to study genetic variation and evolutionary relationships, while protein alignments can reveal conserved functional domains and structural motifs. The choice of sequence type influences the alignment strategy and the interpretation of results. Understanding the nature of your sequences is the first step toward a successful alignment. Multiple sequence alignment is a versatile tool with a wide range of applications. It's used in phylogenetic analysis to reconstruct evolutionary trees, in functional genomics to predict gene function, and in structural biology to model protein structures. MSA also plays a role in identifying disease-associated mutations and in designing primers for PCR experiments. Its applications span basic research, clinical diagnostics, and biotechnology, making it an indispensable technique in modern biology. One of the most powerful uses of MSA is uncovering evolutionary relationships. By aligning sequences from different species, researchers can identify conserved regions that have remained unchanged over millions of years. These conserved elements often indicate essential biological functions. MSA also helps in tracing the origins of gene families and understanding how genes have evolved through duplication, divergence, and selection. Such evolutionary insights are fundamental to comparative genomics and evolutionary biology. MSA is not just about evolutionary history, it's also a powerful tool for functional analysis. By comparing sequences, researchers can identify conserved motifs in domains that are critical for biological activity. For example, aligning protein sequences can reveal active sites or binding regions that are essential for function. This information is invaluable for predicting the function of unknown genes or proteins, and for designing experiments to test their roles in the cell. Multiple sequence alignment has significant applications in disease research. By aligning sequences from healthy and diseased individuals, scientists can pinpoint mutations associated with specific conditions. This approach is widely used in cancer genomics, infectious disease studies, and genetic disorder research. Identifying disease-associated variants helps in understanding disease mechanisms and can guide the development of targeted therapies and diagnostics. Drug discovery is another area where MSA proves invaluable. By aligning protein sequences from different organisms, researchers can identify conserved drug targets and predict how mutations might affect drug binding. This information is crucial for designing effective drugs and for anticipating resistance mechanisms. MSA also aids in the identification of off-target effects, ensuring that new drugs are both effective and safe. Biotechnology, MSA is used to engineer proteins with new or improved functions. By comparing sequences from different organisms, scientists can identify beneficial mutations and design proteins with enhanced stability, activity, or specificity. MSA also supports the development of synthetic biology applications, such as designing novel enzymes or metabolic pathways. Its role in biotechnology continues to expand as new tools and techniques emerge. Forming a multiple sequence alignment involves several key steps. First, you collect the sequences to be aligned, ensuring they are homologous. Next, you select an appropriate alignment algorithm and scoring system. The sequences are then aligned, often using specialized software. Finally, the alignment is evaluated and interpreted, with conserved regions and motifs identified for further analysis. Each step requires careful consideration to ensure accurate and meaningful results. First step in MSA is gathering the sequences you want to align. These can come from public databases like GenBank or UniProt, or from your own experimental data. It's important to ensure that the sequences are homologous, meaning they share a common evolutionary origin. Including non-homologous sequences can lead to misleading alignments and incorrect conclusions. Careful sequence selection sets the stage for a successful alignment. Selecting the right alignment algorithm is crucial for MSA. Different algorithms have different strengths and weaknesses, and the choice depends on the nature of your sequences and your research goals. Some algorithms are optimized for speed while others prioritize accuracy. Popular MSA algorithms include Clustable Omega, Muscle, and MAFFT. Understanding the features of each algorithm helps you make an informed choice for your analysis. Scoring systems are at the heart of sequence alignment. They determine how matches, mismatches, and gaps are evaluated. Common scoring matrices for protein alignments include PAM and BLOSUM, which assign scores based on the likelihood of amino acid substitutions. For DNA alignments, simpler match-mismatch scores are often used. The choice of scoring system can significantly impact the alignment, so it's important to select one that reflects the biological context of your sequences. Gaps are introduced in alignments to account for insertions or deletions. However, too many gaps can distort the alignment, so gap penalties are used to discourage excessive gap introduction. There are different types of gap penalties, such as linear and affine, each with its own advantages. Setting appropriate gap penalties is a balancing act. Too high and you miss real insertions, too low and you get meaningless gaps. Fine-tuning these parameters is key to high-quality alignment. Uncommon approach to MSA is the progressive alignment method. This technique starts by aligning the most similar pair of sequences, then gradually adds more sequences to the alignment. The process is guided by a phylogenetic tree, which helps determine the order of alignment. Progressive alignment is fast and works well for many datasets, but it can be sensitive to errors made early in the process. Understanding its strengths and limitations helps you use it effectively. Iterative alignment method improves upon progressive alignment by repeatedly realigning sequences to optimize the overall alignment. After an initial alignment, the algorithm revisits and adjusts the alignment to correct errors and improve accuracy. This approach is more computationally intensive, but often yields better results, especially for complex datasets. Iterative methods are used in tools like Muscle and MAFFT, making them popular choices for high-quality alignment. Consistency-based methods take a different approach by ensuring that the alignment is consistent across all pairs of sequences. These methods use information from pairwise alignments to guide the multiple alignment, reducing the risk of errors. Tcoffe is a well-known tool that uses this strategy. Consistency-based methods are especially useful for aligning distantly related sequences, where traditional methods may struggle. There are several popular tools for performing multiple sequence alignment. Clustable Omega is known for its speed and scalability, making it ideal for large datasets. Muscle offers high accuracy and is widely used in research. MAFFT provides a range of algorithms for different needs, from fast alignments to highly accurate ones. Tcoffe excels at consistency-based alignment. Choosing the right tool depends on your dataset and research goal. Before running an MSA, it's important to prepare your input data carefully. Sequences should be in a compatible format such as FASTA and should be checked for errors or inconsistencies. Removing redundant or low-quality sequences can improve the alignment. Proper data preparation ensures that the alignment process runs smoothly and that the results are reliable and interpretable. Once your data is ready, you can run the alignment using your chosen tool. Most MSA tools offer both command line and graphical interfaces, allowing you to customize parameters such as scoring matrices and gap penalties. The alignment process may take seconds or hours, depending on the size and complexity of your dataset. Monitoring the process and reviewing logs can help catch any issues early. After the alignment is complete, it's time to interpret the results. Look for conserved regions, motifs, and patterns that stand out. These may indicate functionally important sites or evolutionary conservation. Visualization tools can help you explore the alignment and highlight key features. Careful interpretation is essential for drawing meaningful conclusions from your data. Visualization is a powerful way to explore and present MSA results. Tools like JALView and UGNE allow you to color-code residues, highlight conserved regions, and annotate features of interest. Good visualization makes it easier to communicate your findings and to spot patterns that might be missed in raw data. Effective visualization is an essential part of the MSA workflow. Not all alignments are created equal. Evaluating the quality of your MSA is crucial for ensuring reliable results. Metrics like alignment score, consistency, and coverage can help assess quality. Comparing your alignment to known reference alignments or using benchmarking datasets can provide additional confidence. High-quality alignments are the foundation for accurate downstream analyses. MSA is a powerful technique, but it comes with challenges. Aligning highly divergent sequences can be difficult, and errors in sequence selection or parameter settings can lead to misleading results. Large datasets may require significant computational resources. Being aware of these challenges helps you troubleshoot problems and improve your alignments. There are several strategies for improving the accuracy of your MSA. Using high-quality, homologous sequences, optimizing parameters, and choosing the right algorithm are all important. Iterative refinement and manual inspection can also help correct errors. Regularly updating your methods and staying informed about new tools ensures that your alignments remain accurate and reliable. As sequencing technologies advance, researchers are dealing with ever larger datasets. Handling large numbers of sequences requires efficient algorithms and powerful hardware. Some MSA tools are specifically designed for scalability, allowing you to align thousands of sequences at once. Breaking datasets into smaller groups or using cloud computing can also help manage large-scale alignment. Automation is increasingly important in MSA workflows. Scripting and pipeline tools allow you to automate repetitive tasks, from data preparation to alignment and analysis. Automation saves time, reduces errors, and ensures reproducibility. Many MSA tools offer command line interfaces and support integration with workflow management systems, making it easier to scale up your analyses. MSA is often just one step in a larger analysis pipeline. Integrating MSA results with phylogenetic analysis, structural modeling, or functional annotation can provide deeper insights. Many bioinformatics platforms support seamless integration of MSA with other tools, streamlining your research and enabling more comprehensive analyses. Comparative genomics relies heavily on MSA to compare genomes from different species. By aligning genes or whole genomes, researchers can identify conserved elements, gene duplications, and evolutionary events. These comparisons shed light on the genetic basis of traits, adaptation, and speciation, making MSA a cornerstone of comparative genomics research. Metagenomics involves studying genetic material from environmental samples, often containing many different organisms. MSA helps in identifying and classifying the diverse sequences present in these samples. By aligning sequences, researchers can reconstruct microbial communities, track changes over time, and discover new species. MSA is thus a vital tool in environmental and microbial research. Structural biology, MSA is used to predict the three-dimensional structure of proteins. By aligning sequences with known structures, researchers can infer the structure of related proteins. Conserved residues often indicate important structural or functional roles. MSA thus bridges the gap between sequence data and structural insights, aiding in protein modeling and drug design. Evolutionary studies use MSA to trace the history of genes and proteins. By comparing sequences from different organisms, researchers can reconstruct ancestral sequences, estimate divergence times, and study the mechanisms of evolution. MSA provides the data needed to build evolutionary trees and to test hypotheses about how life has evolved over time. Functional genomics aims to understand the roles of genes and proteins in the cell. MSA helps by identifying conserved domains and motifs that are likely to be functionally important. By comparing sequences across species, researchers can predict gene function and identify candidates for experimental validation. MSA is thus a key tool in annotating genomes and understanding cellular function. Personalized medicine uses genetic information to tailor treatments to individual patients. MSA helps by comparing patient sequences to reference genomes, identifying mutations that may affect drug response or disease risk. This information guides treatment decisions and helps in developing targeted therapies, making MSA a valuable tool in precision healthcare. Agricultural research benefits from MSA by enabling the comparison of crop and livestock genomes. By identifying genes associated with desirable traits such as disease resistance or yield, researchers can guide breeding programs and improve food security. MSA also supports the development of genetically modified organisms with enhanced characteristics. Conservation biology uses MSA to study genetic diversity within and between endangered species. By aligning sequences, researchers can assess population structure, identify unique genetic variants, and inform conservation strategies. MSA thus plays a role in preserving biodiversity and protecting threatened species. MSA is also a valuable teaching tool. By visualizing alignments, students can learn about molecular evolution, gene structure, and sequence analysis. Interactive tools make it easy to explore alignments and understand the principles behind them. MSA thus supports education in bioinformatics, genetics, and molecular biology. The field of MSA is constantly evolving. Advances in algorithms, machine learning, and high-throughput sequencing are driving new developments. Future directions include more accurate alignments for highly divergent sequences, integration with other omics data, and real-time analysis of massive datasets. Staying up to date with these trends ensures that you can leverage the latest tools and techniques in your research. As with any powerful technology, MSA raises ethical considerations. Issues such as data privacy, consent, and the responsible use of genetic information must be addressed. Researchers should follow best practices for data management and ensure that their work complies with ethical guidelines. Responsible use of MSA helps protect individuals and communities while advancing scientific knowledge. A wealth of resources and databases support MSA research. Public databases like GenBank, UniProt, and FAM provide access to millions of sequences. Online tools and tutorials make it easy to get started with MSA, even for beginners. Leveraging these resources can accelerate your research and help you stay current with best practices. The MSA community is vibrant and collaborative. Researchers around the world share tools, datasets, and expertise, driving innovation and progress. Participating in conferences, workshops, and online forums can help you connect with others, learn new skills, and contribute to the field. Collaboration is key to advancing MSA and its applications. Let's recap what we've covered. Multiple sequence alignment is a fundamental tool in bioinformatics, enabling the comparison and analysis of biological sequences. It has wide-ranging applications in research, medicine, agriculture, and more. By understanding the principles, methods, and challenges of MSA, you can harness its power to advance your work and contribute to scientific discovery. Thank you for joining us on this journey through multiple sequence alignment. Whether you're a researcher, student, or enthusiast, mastering MSA opens up new possibilities in understanding life at the molecular level. Continue exploring, experimenting, and learning. Bioinformatics is a rapidly evolving field, and your curiosity will drive future discoveries. Stay tuned for more in-depth explorations of bioinformatics tools and techniques.